All right, today we're tackling a fascinating condition, multiple myeloma. It's a story about what happens when one of your body's own defenders decides to go rogue. I mean, think about it. Your immune system is this incredible, highly trained army. So what happens when one of its own soldiers just rebels? Let's trace that chain reaction, because it's pretty wild. So what is this stuff? Well, at its heart, multiple myeloma is a cancer of your plasma cells. And you can think of these plasma cells as these amazing little factories inside your bone marrow. Their job? To pump out all sorts of different antibodies to fight off infections. But with myeloma, one of those factories just goes completely haywire. It starts turning out just one kind of antibody, a totally useless one, over and over again. Okay, so here's our game plan. We're going to start at the very beginning with that single cell going rogue. Then we'll look at the chaos it causes using this really handy acronym CRAB. After that, we'll put on our detective hats to see how it's diagnosed, check out some of its close look-alike relatives, and wrap up with the path forward. All right, let's dive into section 1. We're going right back to the beginning, the origin story of this whole mutiny. So normally, this is how it's supposed to work, and it's actually a beautiful system. You have these things called B cells, a type of white blood cell, and they grow up to become plasma cells. These are the specialists, right? They produce this huge, diverse arsenal of what are called polyclonal antibodies. Poly meaning many, so they come from all different cell lines, ready to take on pretty much any invader you can think of. But with myeloma, yeah, that whole system just breaks down. A single plasma cell mutates. And this one rogue cell, it just starts cloning itself over and over and over. And this new army of clones, they all produce just one type of antibody, a useless monoclonal protein. The assembly line analogy is perfect. It's like it's stuck, just stamping out thousands of copies of the exact same broken part. So, what's actually kicking off this rebellion? Well, it often starts with a specific genetic hiccup, like something called a 13q deletion. You can think of that as a faulty off switch on the cell's DNA. Then you get this chemical messenger, interleukin-6, which acts like gasoline on the fire, just screaming at these rogue cells to multiply. So you have this genetic mistake plus the growth signal, and that's the 1-2 punch that really gets this disease going. Okay, so this rogue army is growing out of control. What happens next? Well, now we're going to see the damage it does across the entire body. And to get a handle on the major symptoms, there's this really clever and frankly easy to remember acronym that doctors use, CRAB. Each letter represents one of the 4 classic telltale signs of myeloma. First up, C is for calcium. So these cancer cells start pumping out a protein, MIP-1 alpha, that basically wakes up your body's bone-demolishing crew, the osteoclasts, and tells them to go into overdrive. They start tearing down bone, and that releases this huge flood of calcium into your blood. That's a really dangerous condition called hypercalcemia. Next, R is for renal failure. That means kidney failure. And this is a perfect example of how one problem just snowballs into another. First, all that extra calcium floating around from the bone breakdown is really hard on your kidneys. But that's not all. Those useless antibodies, they're made of smaller parts, and these tiny bits called light chains start to literally clog up the kidney's filtering system, like gunk in a drain. Okay, on to A, which stands for anemia. Remember our little factories in the bone marrow? Well, at this point, they are completely overrun. They're just crowded out by all these cancerous plasma cells. There's literally no more room at the inn to make healthy red blood cells, the ones you need to carry oxygen. And that's what leads to anemia, which is why people feel so tired and weak. And finally, we have B for bone lesions. That same process that's jacking up the calcium, those out-of-control osteoclasts, is also literally dissolving parts of the bone. It creates these weak spots, these lesions. And on an X-ray, they look just like someone took a hole punch to the bone. You see these punched-out holes most often in the vertebrae of the spine and in the skull. So with all this damage happening all over the body, how do doctors nail down myeloma as the actual culprit? Let's get into the investigation. It really boils down to this magic number. The key test is a bone marrow biopsy. And if doctors find that more than 10% of the cells in that sample are these clonal cancerous plasma cells, and the person has at least one of those CRAB symptoms we just talked about, boom, that's the confirmation. That's a diagnosis of multiple myeloma. But wait, how do they actually tell the good cells from the bad ones under the microscope? Well, they look for specific markers on the surface of the cell, kind of like a team jersey or a uniform. Healthy plasma cells wear markers called CD19 and CD45. The cancerous ones? They've ditched their jerseys. They're CD19 and CD45 negative. It's a dead giveaway. And the investigation doesn't stop there. The blood and urine turn up other classic clues. You'll see something called an M spike. It's this huge, sharp peak on a graph that just screams, one protein is being mass-produced here. You also see this weird thing called rouleau formation, where red blood cells start stacking up on each other like coins because of all the extra protein in the blood. And finally, those little light chains that clog the kidneys, they spill into the urine, where we call them Bence-Jones proteins. All right, section 4. Now, it's important to know that myeloma doesn't exist in a vacuum. It actually has a few close relatives, conditions that can look really similar at first. So telling them apart is absolutely critical. So let's look at the whole spectrum here. Before you get to full-blown myeloma, you often have these precursor stages. Think of MGUS as the earliest, mildest form. You've got a few rogue cells, but not many, and zero symptoms. Then there's smoldering myeloma, which is the next step up. The numbers are higher, the rogue cells are multiplying, but, and this is key, the crab symptoms haven't kicked in yet. What really tips a diagnosis into active multiple myeloma is the presence of that damage, the crab symptoms we talked about. And here's another important lookalike to distinguish, Waldenström's. Okay. Both of these cancers involve a rogue antibody clone, right? But here's the difference. Myeloma usually makes smaller antibodies, like IgG or IgA, and it's famous for causing those bone lesions. Waldenström's, on the other hand, makes this huge, clunky antibody called IgM. This giant protein makes the blood really thick and syrupy. But, and this is a huge but, it does not cause those punched-out bone lesions. So the diagnosis is confirmed, we know it's not one of its relatives. What's next? What does the path forward look like? Well, first things first is staging, and to figure out the prognosis, there is one single factor that is more important than anything else, the level of a protein called beta-2 microglobulin. It's pretty simple. The lower that number, the better the prognosis, the earlier the stage. It's basically a direct measure of how much cancer is in the body. And what about the battle plan? How do we treat this? The modern approach is really a multi-pronged attack. You've got these targeted drugs, lenalidomide and bortezomib, that go right after the cancer cell's survival machinery. You combine those with a powerful steroid like dexamethasone. That combination is really the backbone of modern treatment because it attacks not just the rogue cells themselves, but also the environment that's helping them thrive. Which brings us to a really interesting final thought. Remember, this whole journey started with a single cell, with one tiny mutation. And as science gets better and better at identifying the exact genetic triggers that cause this, the ones we touched on earlier, it really makes you wonder, right? Could the future of treating this disease be less about just managing the fallout and more about finding that one broken genetic switch and just flipping it back to off?